Hi, I am Dr. Ranjit Chauhan. The title of this review is Earliest HBV Post-Genome Integration, Sites, Mechanism, and Significance in Carcinogenesis. This review is accepted for publication in Hepatoma Research under Creative Commons license. I would like to draw your attention to the table given at the left-hand side. Four independent studies using five different HBV infection systems showed that HBV can integrate from 15 minutes onwards up to 24 hours post-infection. Most of the genes into which HBV integrated were of translocation potential as well as retrotransposon genes. The type of junctions which were formed between the virus DNA and host DNA were head-to-tail junctions and in a few by overlapping homologous junctions. At the right-hand side is an example of head-to-tail using non-homologous-end-joining junction is given. We also uncovered the mechanism of HBV integration. At the left-hand side of the column, at x-axis the time points from minutes to hours are given, and on y-axis the components of virus machinery, oxidative stress, and host repair machinery is given. As you can see, HBV integrated much earlier before the cccDNA and RNA was detected, and reactive oxygen species and DNA damage was detected immediately after the HBV infection, whereas reactive nitric oxide and antioxidant repair machinery hemoxygenase was also detected. In host DNA repair machinery, PARP1, XRCC1, OG1 were detected at different time points, which are given here. And at right hand side of the column is a cartoon picture showing the mechanism of HBV integration by non-homologous end joining. To summarize the results presented in this review, HBV can integrate as early as 30 minutes post-infection of HepG2 NTCP cells and 1 hour post-infection of HepRD cells. Not only this, using the in vivo woodchuck animal model, we identified that WHV can integrate up to 3 hours post-infection. The frequent genes into which HBV integrated were retrotransposon genes and genes with translocation potential. The type of junctions which were formed were of non-homologous end joining, implying that PARP1 and XRCC1 dependent repair machinery is working. We also identified that oxidative stress is a culprit in this process. Thus, we can conclude that HBV can engage the mobile genetic elements and genes with translocation potential right from the beginning of the infection, and thus causes the pro-oncogenic perturbations throughout the genome. Thank you.